Hello, Namaste. In this video, we are going to deal with the silicon technology for the management of the major pest of wheat, that is pink stem borer, Sysamia inferens. Here, we are going to deal with the different sources of silicon for the management of pink stem borer under field conditions. Pink stem borer, Sysamia inferens, is an emerging pest of wheat in northern plains and central parts of the India. In early stages, it causes dead odds, and in later stages, that is in reproductive stages, it causes white ear rates. It is difficult to control with the use of insecticides because of its peculiar nature of feeding inside the stem. Silicon is the second most abundant element in the earth crust, which is nearly everywhere available and is taken up by the plants in its soluble form of silicic acid. Soluble form of silicic acid, which is also known as the monosilicic acid, is the plant available portion of the silica that makes up over 40% of the soil chemistry. It is generally present in plant tissues in amounts similar to that of the macronutrients. Like, we have conducted an experiment to evaluate different sources of silicon along with potassium to manage the pink stem borer in wheat. One source was the soil applied source, that is diatomaceous earth at 150 and 300 kg per hectare, and another source is foliar applied silicon source, that is orthosilicic acid at the rate of 2 ml and 4 ml per liter. And potassium was evaluated at 18 kg and 36 kg per hectare. We have noticed that all silicon sources significantly reduced the pink stem borer damage and significantly improved the photosynthesis, yield, and related parameters in wheat under field conditions. Among silicon sources, diatomaceous earth at the rate of 150 kg per hectare was significantly better treatment among other treatments and significantly improved the uh, photosynthesis and related parameters. We have noticed that the silicon along with potassium able to activate defense enzymes in wheat plants in against pink stem borer incidence, and it reduced the impact of pink stem borer in wheat conditions. So it is noticed that the silicon is able to activate peroxidase, phenylalanine ammoniolase, polyphenol oxidase, and beta 13 glucanase, and so on. In continuation of this study, now we are focusing on use of nanosilicon for the management of pink stem borer in wheat under field conditions to minimize the application rates of diatomaceous earth, that is